Okay, so we are going to talk about this drug cisplatin. Okay, so I've got two of the bases, DNA bases, adenine and guanine there, which we'll see why I've got there in a minute. But cisplatin is it's a complex ion. You've got a central platinum ion, and it's got two ligands. It's got two chloride ions and two ammonias. Right, now that is the form I've drawn there. It's called cisplatin, and that is the active drug. The reason why it's called cisplatin, well, cis is the old-fashioned way of saying Z, as in the Z geometric isomers, and you've got the chlorines both on the same side there, or you can think of the ammonias on the same side. Transplatin, which is not as an effective a drug at all, would be, so you'd have one ammonia there and the other ammonia opposite it. Okay, right. The shape of this is it's not tetrahedral. You've only got four ligands. It's actually square planar. So all the bond angles are 90. The chloride ions have a charge of -1. And the overall cisplatin is uncharged, so that means the platinum must have an oxidation state of 2 there. It's got a charge of 2+. Okay. Right. So how does it? It will kill, preferentially. It will kill cancer cells. And how does it do that? Well, it does that by essentially it will form ligands. The guanine residues in DNA can act as ligands to the central platinum there. Okay, so if you remember, that nitrogen is the one that is attached to the deoxyribose sugar. Adenine I've put here as well because it does bind. It will bind to adenine to some extent, but not as well as guanine. So that's where that's attached to the deoxyribose. And it's this nitrogen here and this one here, the lone pair on that. So I'll draw the lone pairs on them. Okay, they can form the dative bond to the platinum in the cisplatin complex ion. Right. So very briefly, then what happens? So you have cisplatin. So that's PT. Cl2 NH3 2. When it enters the cell, one of those chlorine ligands is replaced by a water, and that's because the chloride ion concentration is lower in the cell than it is in the extracellular fluid. So that will form PtCl H2O NH3 twice, and you'll get a chloride ion coming off there. And this species, this can then bind to the DNA. So that will form, it will bind to the DNA because the guanine forms a dative covalent bond to it. So when it does bind, what happens is, so when it first binds, the H2O ligand is replaced by a guanine residue in the DNA. So the guanine won't be free guanine, obviously it will be bound to ribose and part of the DNA double helix. And then what happens then is the Cl, one of the remaining Cl minus, is also replaced by a guanine. So the kind of reaction that's going on there is it's a ligand exchange reaction. So just like you have, you know, you can, if you get copper hexaqua with 6 waters around it, and you add conch ammonia, you will replace some of those waters with NH3. It's the same sort of reaction there, ligand exchange. Now, why does that cause DNA damage? It causes DNA damage mostly. So here's going to be a piece of DNA, and I'm just going to put some random bases like A, C, G, G, T, A. So there's a piece of DNA. Right, what it will do is the platinum will form. So if I draw my cisplatin here, I've still got the 2 ammonia ligands attached. And well, these are all dative bonds, I should put arrows on them. There's your cisplatin complex. When you've got 2 Gs next to each other like that, it's particularly damaging to the DNA, because that cisplatin will be bound to the DNA, and then for DNA to replicate, you probably know that it needs to unzip. So, and the enzyme has to bind to the strand, and of course it won't be able to do that if this big cisplatin thing is in the way. So it will stop DNA replication. The cell tries to repair the DNA, and when it can't do that, the cell undergoes programmed cell death. So it basically kills itself, which is called apoptosis. If you do biology, you probably heard about apoptosis, right? But it basically works by stopping DNA replication. So you may think, well, why doesn't it kill healthy cells? Well, it does to some extent, but not to the usually malignant cells, cancer cells, are dividing more rapidly. So most of the toxic effects on cells will be concentrated in the cancer cells and hopefully not in the healthy cells. But of course, it does damage the healthy cells also, and that causes all the side effects that are associated with chemotherapy. I think they've asked in the past paper what can be done to assuage the toxic effects of the chemotherapy, and I think the answer in the mark scheme says so the drug is given in repeated cycles. So it's not given in one huge dose, it's given in several repeated smaller doses. Right, so that's all you need to know about cisplatin. It kind of crosses over, really. It's in the biochemistry section, but it really crosses over with the transition metal topic in the inorganic part of the syllabus. I think section 2.5, I think it is. Yeah, 2.5. Okay.